Hi everyone, I'm Zixia from CSBL. Today my presentation title for the journal club is Mycena Genomes Resolve the Evolution of Fungal Bioluminescence, which published on PNAS on 2020. Bioluminescence is a common natural phenomenon found in bacteria, insects, and fungi. Among the 81 known bioluminescent fungi, at least 68 species belong to Mycena, yet they account for less than 12% of the approximately 600 Mycena species. In this study, they selected 5 Mycena species, 4 of which are bioluminescent and 1 that is non-bioluminescent. As shown in figure, these species exhibit similar morphology but differ significantly in their bioluminescent capability. The figure illustrates the proposed pathway for fungal luciferin biosynthesis and recycling. In this process, histidine synthesis converts caffeic acid into histidine, which is then hydroxylated by H3H to form 3-hydroxyhistidine. Luciferase catalyzes the addition of molecular oxygen, generating a high-energy endoperoxide intermediate that decomposes, producing oxyluciferin and emitting light. Oxyluciferin is then recycled back to caffeic acid through the action of caffeine pyruvate hydroxylase. So, how did the bioluminescent genome originate? Why have some Mycena species lost their bioluminescent ability? Besides the known bioluminescence-related gene, what other genes might be involved? To address this question, this study systematically analyzes the genomes of Mycena species and constructed their phylogenetic tree to trace their evolutionary history. The figure presents the species phylogenetic tree, constructed based on 360 single-copy orthologous genes. Phylogenetic analysis indicates that the genus Mycena and the genus Armillaria share a common bioluminescence ancestors and began to diverge in the late Jurassic, approximately 160 million years ago. Blue dots in the figure represent high-confidence branches in the phylogenetic tree, while the green bar chart indicates the proportion of bioluminescent species. Notably, although the proportion of bioluminescent Mycena species is high, not all species exhibit bioluminescence, suggesting that bioluminescence-related gene might undergo loss or recombination. In Figure 1c, we observe variation in copy numbers of gene families related to luciferin biosynthesis across different Mycena species. Significant differences are evident in the 3 core genes, LUZ, H3H, and CPH among species. For instance, some non-bioluminescent Mycena species have fewer copies or even a complete absence of these genes, implying that their loss might have led to the loss of bioluminescent capability. This observation further supports the hypothesis suggested by phylogenetic tree that bioluminescence is not a conserved trait in all Mycena species. Figure 1d displays the results of phylogenetic reconciliation, showing that the author group containing luciferase was present in the last common ancestor of the fungal mycenoid and marasmioid lineage, and the family Schizophyllocraceae, predating their incorporation into the luciferase group. From Figure 1e, we can observe the genome size and the proportion of repetitive sequences in Mycena species, including LTR, LINE, SINE, DNA transposome, and so on. The accumulations of these repetitive sequences may influence genome plasticity, and DNA methylation plays a crucial role in regulating their activity. Figure 2A to C further analyzes the relationship between gene density, transposable elements distribution, and DNA methylation levels. The results show that regions with higher DNA methylation levels are often rich in transposable elements but have lower gene density. In Mycena, CG methylation levels in genes are significantly lower than in repetitive sequences. Species with larger genomes and higher repetitive content, such as Mycena sanguinolenta and Mycena venus, exhibit lower methylation levels in repetitive sequences. In contrast, species with smaller genomes, including Mycena indigotica, Mycena chlorophyllus, and Mycena contingensis, show much higher methylation levels in repetitive sequences. These findings suggest that variation in repetitive sequence composition is mediated by differences in DNA methylation among closely related Mycena species. The figure illustrates genome synteny among different Mycena species. Lines between scaffolds represent single-copy orthologs between species. In Mycena, multi-genome comparisons reveal that synteny conservation is often lost at scaffold end or extends several megabases in assemblies. Clustering of orthologs genes divides scaffold into low synteny and high synteny region. The study indicates that highly synthenic regions in Mycena are typically located at scaffold center. Further analysis examines synteny around the luciferase cluster among bioluminescent fungi. Most Mycena luciferase clusters contain AUZ, H3H, CYP450, and Hispase. However, synteny is absent among genes surrounding the luciferase cluster in mycenoid and Amplotus lineage species. This suggests that the luciferase cluster, located in a rapidly evolving genomic region, may be prone to frequent loss, possibly mediated by transposon-driving rearrangement. In Mycena, the luciferase cluster within the mycenoid lineage is located in a highly dynamic genomic region with low synteny, making Mycena fungi more susceptible to losing the luciferase cluster. In contrast, the luciferase cluster in Armillaria resides in a slow-evolving region, making it more conserved. This pattern suggests that the loss or retention of bioluminescence-related gene depends on dynamic changes in genome structure. A total of 29 orthogroups contain upregulated genes members across all four Mycena species, including LUZ, H3H, and Hispase, which correlate with bioluminescence intensity depending on their expression within the luciferase cluster. Moreover, significant differences in bioluminescence gene expression are observed across different growth stages and tissues. These findings indicate that the regulation of bioluminescence genes is influenced not only by evolutionary factors, but also by developmental stages and tissue specificity. The bioluminescence in Mycena fungi originated from a single emergence of the luciferase gene cluster, but this cluster has undergone widespread loss during evolution, leading to a discontinuous distribution of bioluminescence among different species. The luciferase gene cluster is located in a low synteny region, making it more susceptible to genomic rearrangements and loss, which contributes to variation in bioluminescence capability among different Mycena species. Gene expression analysis shown that bioluminescence gene exhibits tissue and stage specific expression in Mycena species, suggesting that their regulation may be influenced by environmental and developmental signals. The ecologic role of fungal bioluminescence remains inconclusive. It may be involved in attracting insects for spore disposal or serving specific physiological adaptation to environmental conditions. That's all. Thanks for your listening.